Welcome to The Explainer. Today, we are taking a slow, deliberate look at one of the absolute most remarkable cells in human biology. You know, if you've ever looked into IVF, you might just naturally assume that retrieving an egg is the main hurdle. But getting the egg is actually only the first step. The true challenge, and honestly where the science gets incredibly fascinating, is how we read it. Because an egg can be retrieved, look totally intact, and still not be ready for fertilization. So what exactly are embryologists looking for once that egg is safely in the lab? Well, that is exactly what we are going to explore today.
Okay, let's dive into this. We're going to walk right through our chronological lab workflow to see exactly how an embryologist assesses these vital cells. We'll start with why counting isn't enough, then look at anatomy and denudation, break down the maturity stages of GV, M1, and M2, check out morphology, and finally, tackle the really tricky part, appearance versus true competence.
All right, part one, counting is not enough. This is our initial reality check in the lab.
When we hit that first major clinical filter on retrieval day, there's this really common myth that every single retrieved egg is automatically usable. But unfortunately, that's just not the reality. On retrieval day, you might hear a doctor say, "Great news, 10 eggs were collected." That recovery count is fantastic, but it really just tells you how many eggs were physically found floating in the follicular fluid. For the embryologist, the real work begins immediately with one massive question: how many of those retrieved eggs are actually developmentally ready? We absolutely must identify this mature fraction first, because only those specific eggs are ready for the next step of fertilization.
Before we go any further, let's just make sure we've got our terminology locked down. In the lab, we don't usually just call it an egg. We use the specific term oocyte. An oocyte is a large, highly specialized female reproductive cell. In fact, it is the single largest cell in the human body. It's just barely visible to the naked eye. Grounding ourselves in this specific word, oocyte, is important because, as we're about to see, this single cell is structurally a lot more complex than you might think.
Moving right along to section 2, anatomy and denudation. Or, as I like to think of it, stripping away the noise. So, when an embryologist first looks at an oocyte under the microscope, they don't just see a simple round sphere. They actually see a complex series of boundaries and layers. It's almost like a tiny biological fortress. On the very outside, you have this fluffy cloud made up of cumulus and corona cells. Beneath that cloud is the zona pellucida, which is this tough protective outer shell. Inside that shell is the perivitelline space. Basically, a small fluid-filled gap before you hit the main core of the cell itself, called the ooplasm, or cytoplasm. And finally, there's the polar body, which is a tiny cellular structure that, when it's there, acts as a massive visual marker of maturity. These features are essentially the visual language an embryologist uses to read the oocyte.
Let's move to and see how this builds, because to actually see those internal structures clearly, which is absolutely required for a fertilization method like ICSI, the embryologist has to perform a really delicate procedure called denudation. First, the oocyte gets a brief exposure to an enzymatic media, usually something called hyaluronidase. Second, the embryologist uses incredibly gentle mechanical pipetting. And third, this combination carefully strips away all those outer cumulus and corona cells. It's a highly delicate dance, honestly, because overhandling can easily damage these incredibly fragile cells. And this brings us to a really vital realization about the lab process. Denudation is not just a basic cleaning step. It is the defining moment when the oocyte is laid bare and becomes fully readable to us. Without stripping away that dense outer cloud of cells, an embryologist literally can't accurately assess the internal structures, and they certainly can't plan for sperm injection. It's the exact moment the egg finally reveals its true developmental stage. Okay, section 3. Let's talk about those maturity stages we keep mentioning, GV, M1, and M2. In the lab, we classify maturity incredibly methodically. First up is the GV, or germinal vesicle stage. This is a totally immature oocyte. Visually, you can actually see the nucleus sitting in there. It looks like a distinct little bubble, the germinal vesicle. Is it ready for ICSI? No, definitely not. Next, the cell progresses to the M1 stage, or metaphase 1. At this point, the germinal vesicle has broken down, so the bubble is gone, but there is still no polar body visible. It's a transitional state, but it's still immature for routine fertilization. Ready for ICSI? Again, no. Finally, we reach the M2 stage, or metaphase 2. The defining visual marker here is the presence of that first polar body tucked right into that perivitelline space we talked about earlier. Is it ready for ICSI? Yes, absolutely. Which leads perfectly to this wonderful summary from the source material. In practical IVF terms,
M2 is the egg the embryologist is hoping to see. This M2 stage is, quite literally, the holy grail for ICSI. It represents an oocyte that has successfully completed the key meiotic progression it needs to be ready for fertilization. Seeing that M2 is the ultimate goal of this entire maturity checkpoint.
But that raises a pretty pivotal question, doesn't it? One that every single fertility professional grapples with daily. If an oocyte has a polar body, and we officially classify it as an M2, is it perfect? Does reaching maturity guarantee that it's a flawless egg?
Well, that brings us to section 4, morphology and dysmorphologies. Basically, visualizing the quality of the cell. Now, what's really interesting about this is the sheer variety of visible quirks, abnormalities known in the lab as dysmorphisms, that embryologists are constantly on the lookout for once the egg is denuded. Even a fully mature M2 oocyte might have dark or really granular cytoplasm instead of a nice, smooth, clear texture. It might have vacuoles, which look like little fluid-filled cysts right inside the cell. Sometimes there's debris trapped in the perivitelline space, or maybe an abnormal, super thick zona pellucida. We even see irregularly shaped or totally fragmented polar bodies. Embryologists document all of these features very, very carefully. But here is a critical nuance you really have to take away from this explainer. Oocyte morphology gives us clues, not certainties. These visual findings are just descriptive. They are absolutely not a final verdict on the fate of that egg. While some dysmorphisms definitely have been associated with poorer outcomes, their predictive value can be wildly inconsistent. It's crazy, but some highly unusual-looking eggs can still fertilize perfectly and turn into beautiful embryos, while some perfectly normal, textbook-looking eggs might fail entirely. Which segues right into our final section, appearance versus true competence, the humbling truth of the IVF lab. This concept really highlights the gap between what is visible to the human eye and what is biologically real. On one hand, we have nuclear maturity. This is the visual readiness, looking through the microscope, seeing that polar body, and confirming, yep, it's reached the M2 stage. But on the exact opposite side of the coin, we have cytoplasmic competence. This is the internal, completely invisible molecular readiness of the egg to actually support fertilization and drive embryo development. An oocyte can look completely, flawlessly mature on the outside, but entirely lack the hidden internal machinery needed to actually create an embryo. So what actually influences that invisible cytoplasmic competence? If you've ever wondered why IVF results can vary so wildly from cycle to cycle, well, it's because of this delicate web of variables. Factors include maternal age, exactly how the ovaries responded to stimulation meds, the precise timing of the hormonal trigger shot, and of course, the lab handling conditions. But perhaps the most fascinating factor is just intrinsic biologic variability. Even within one patient, from a single retrieval, you get a mixed bag of eggs. They are not all equivalent, and their internal, hidden quality varies drastically.
So the crucial point is summarized right here. The lab can identify maturity more easily than competence. It's a very humbling limitation. We have the fancy microscopes and the precision tools to see if an egg has reached the M2 stage. We can log every single visible dysmorphism. But the true functional competence of that cell, that remains largely biologically hidden from us until we actually attempt fertilization and watch how it develops over the next few days.
And that right there is why this thorough assessment process matters so much. A high retrieval number is a great start. An awesome start, really, but it's only a start. The entire downstream IVF process, the fertilization, the embryo culture, and ultimately achieving a pregnancy does not move forward on egg count alone. It moves forward entirely on mature, functionally competent oocytes. Which leaves us with one final kind of provocative thought. We've spent this entire time learning exactly how to read the egg, but the egg is really just one piece of the puzzle. Since an M2 oocyte only gives us half the picture, what does it take to prepare the other half of the fertilization equation? How do we assess and prepare the sperm to meet this incredibly complex cell? Well, that is the fascinating science waiting for us in the andrology lab. Thank you so much for joining me for this explainer, and keep exploring.